Thanks, Ramona, and thanks for everyone for your questions and the discussion. As my colleague said, we will now come to the second block of the webinar, which will focus on bioleaching. And the first presentation will be held by Päivi Kinnunen. He's a research professor in circular economy of energy metals at VTT in Finland, and an adjunct professor in process technologies for circular economy at Tampere University. Her research includes industrial circular economy, valorization of mining industry side streams, and bioprocess development and closing water loops. She holds a Doctor of Science in Technology and a Master of Science in Economics and Business Administration. And
good afternoon, everybody. And like said, this is going to be the general presentation about bioleaching, and then the next presentations are going to be more technical ones. And I have a really bold question. So can microorganisms revolutionize metal recovery? And let's see what is the answer to this. Okay, now I'm having trouble in getting the presentation to go forward. So let's see. I guess it's now sharing. Are you able to see the slides?
Yes, we can see it. And
okay, great. So basically, this is the very simplified principle of bioleaching. So what happens? So we have different kind of iron and sulfur oxidizing microorganisms, and they are able to oxidize iron to ferric iron, and then they are able to oxidize different kind of sulfidic minerals. And in this way, they are producing ferric iron that is a leaching agent, and they are also producing acidic environment. So basically then these leaching agents are leaching the metals from the mineral so that we get the metals into the solution, and then we have a leach residue. And yeah, so this is a very simplified picture of that. And this is the history of bioleaching and what has happened there. So it has been already used for hundreds of years or even thousands of years, but there was like no idea of microorganisms that they are involved. So it was only considered as something occurring naturally. And only in the 1947, the role of microorganisms was demonstrated. And this was actually the point where it's possible to optimize the conditions for microorganisms when you know that it's based on their role. And a few years later, then Thiobacillus ferrooxidans and Thiobacillus thiooxidans were identified, and later they were named acidithiobacillus. And the first bioleaching operations were dump leaching, later heap leaching. In the 1980s, it was going forward to reactor leaching. And then I mention here specifically the heap leaching in Finland, because it was the one that was taking place in Europe. And this means that the different bioleaching technologies are commercially used worldwide. And there have been also estimations how big portion of different metals is produced using bioleaching. And the estimation is that about 10 to 20% of copper is produced globally using bioleaching, perhaps about 1% of gold, and then small amounts of other base metals such as cobalt and nickel are mentioned as well. So typically the base metals are recovered with heap leaching technologies, and then gold is recovered with reactor technologies. And like I said, we have different technologies from reactors to vat leaching, to dump and heap leaching, and even to in-situ leaching. And in reactors, it's typically what we use the concentrates. There is also one case about coupled tailings processing there. It's easier to control different parameters in the reactor. The leaching times are short from hours to days, and the costs are then higher. Then in comparison to the other leaching technologies, these are merely for the lower grade ores, very low grade materials, but then it's more challenging to control the parameters, and then the leaching times are much longer, meaning from months to even years. But the costs are much lower than with reactors. There was one question when this heap leaching operation started in boreal conditions, that is it possible to maintain the temperature within the heaps if in the boreal conditions the temperatures in the winter time can be even -40 degrees. And this has been now proven that the temperature is maintained by exothermic leaching reactors also in the boreal conditions. So this is a sign that it can be used globally, worldwide. The first top figure is about these commercial operations for sulfidic ores. And like I explained earlier, that these reactions are based on the iron and sulfidic mineral oxidation reactions. So we are using iron oxidizers and sulfur oxidizers in these processes. And this is basically the process that is used in commercial operations. On the other hand, there are also some different kind of reactions that are in the research phase for bioleaching. So it's also possible to use the bioleaching in the reverse mode. So it's possible to use the iron reducing microorganisms to leach certain metals out. On the other hand, we also have certain kind of microorganisms that are able to produce organic acids out of organic compounds, and these organic acids can be used for leaching. And then there is also the case that we have a certain kind of cyanogenic bacteria and fungi that are able to produce cyanide that can be then used for leaching. But like I said, these are more in the research stage. So then, of course, one of the questions is that where to find these bioleaching microorganisms. And this is one example. So, of course, one of the best places is to go to the mine sites, because there the microorganisms have been already adapted to the right kind of conditions. And this is an example of one underground mine where we were taking samples, and it's also quite obvious that there is iron oxidation going on, and there were like a lot of good iron oxidizing microorganisms that can be used in bioleaching. So what are the advantages of bioleaching? So it's considered more environmentally friendly, but of course it's not always the case. So it needs to be really like calculated, and we need to do the LCA for that. But there are like no sulfur dioxide or other harmful gas emissions there. And it has been shown that there is like less electricity and energy requirements. So the bioleaching based production in Finland, a third party has calculated that it consumes about 90% it's less electricity and thermal energy, and then it has about 60% its low carbon dioxide footprint in comparison to the industry average. So it's pretty simple to operate and maintain, and the residues are chemically less active. And also we are not needing any high pressure or high temperature, so the operation conditions are pretty mild. Of course, then there is the issue that it takes a lot of time. So that is one of the challenges of bioleaching, that it definitely takes more time than with chemical leaching. But this is basically the short introduction to bioleaching in general, and now we have the technical presentations that go more into the details of bioleaching processes. And perhaps, like I said, can bioleaching revolutionize the metal recovery? It's like, on the other hand, it has already done so. So quite big part of copper is recovered by bioleaching, but on the other hand, it's still a niche technology. It's suitable for certain material streams, but it's not one solution for all different challenges. Okay, thank you.
Thank you very much. Our next speaker is Tiina Heikola. She's a research scientist at VTT and a PhD student at Tampere University, and her background is in analytical and inorganic chemistry. She has been working with metals and material recovery topics now about 7 years in the field of hydrometallurgy. And at the moment, she's doing her PhD studies on selective recovery of critical metals from low-grade materials using bioleaching and chemical precipitation. Yeah, Tiina, please go ahead.
Thank you, Jana, for the introduction, and good afternoon from my side as well. Yes, so my name is Tiina Heikola, and I will be talking about bioleaching and how we can exploit it in the METALLICO, or how we have exploited in METALLICO project, and more specific in the task where we are developing the COMAN process. My presentation will not concentrate maybe so much on the reactor design, but more on the practical side, what we have been doing in this project or in the METALLICO project, and how we have developed the bioleaching process. And I think my presentation, because I will start shortly going through the 5 METALLICO processes. So we have 5 different processes: Cool+, Tailco, Purchase, CONI, and COMAN processes that we are developing in the METALLICO project to recover battery-grade lithium carbonate and cobalt sulphide, as well as cobalt, copper, manganese, and nickel concentrates. And the main objective in the METALLICO project is to kind of demonstrate that we can process and upscale these processes sustainably in different industrial sites, from lab scale to industrial relevant environment. And as I said, we have been working in COMAN process, and I will shortly go through the COMAN process in stepwise. So in the COMAN process, we are working on the recovery of cobalt, copper, manganese, and nickel from the tailings produced in the sulfuric acid production plant. And the process is divided in 2 separate steps: pre-treatment and then hydrometallurgy. And in the pre-treatment stage, Tarsis Mining Company, with support from UPC from Barcelona, who are our partners in the project from Spain, they are working to separate and concentrate the target metals from the initial material through mechanical separation and dual flotation. And then the concentrates from this pre-treatment phase are leached in the hydrometallurgical phase, followed by the impurity removal and selective sulfide precipitation. And at the end, the leaching residues will be sent back to Tarsis for valorizing of the remaining zinc. This is maybe a little bit repeating what Päivi already explained or presented in the previous presentation, but I will shortly go through this slide. So what is bioleaching and some practical approach to it? So in bioleaching, we are exploiting microorganisms to extract metals from ores or metal-containing wastes by chemical reactions they would naturally carry out. The aim is to provide oxidizing and acidic environment where minerals dissolve and the metals are released into the solution. And in the practical way, in simplified terms, the activity of the microbes and as a result of that, the leaching success can be monitored through pH and redox potential evolution. And as Päivi already mentioned, originally the bioleaching is evolved from a simple dump leaching, but nowadays it can be even a biomolecular level engineered process. And it is usually considered ecological and economical alternative to conventional techniques. The advantages are reduced energy and chemical consumption, which are particularly an advantage when treating low-grade materials. And now I will move on to the kind of process development in the METALLICO project and like concentrating on the bioleaching. So we started the work with the sampling trip to a closed mine site in central Finland, and we collected samples from different depths and did the microbiological analysis afterwards in the lab. The microbes were first cultivated and then adapted to a new material gradually. And after the adaptation, multiple different leaching tests were performed, both chemical and biological, in the small laboratory tests. And based on those results, bioleaching was selected as the leaching method for the COMAN process. Then we continued the work with the several small batch reactor tests in 1 liter glass reactors to study the effect of different parameters, including temperature, solid liquid ratio, pH range, as well as the chemicals for the pH adjustment, possible additives and catalysts to enhance the leaching efficiency and rate, and also recycling of the bioleaching leachate. In general, as already mentioned, bioleaching is a slow process compared to, for example, chemical leaching, and the experiment time is measured in days or even weeks rather than in hours. And in the case of bioleaching, parallel tests are also very essential because the system is variable, adaptive, and non-linear, and without replication, it is difficult to know whether the result reflects the process itself or is it just one biological trajectory. As a conclusion, testing all of these different parameters and their combinations took a huge amount of time, and I would say that it was maybe the most laborious part of the whole whole work or the process development to test all the different parameters and their combinations. Then we moved to a larger scale in 5 to 10 liter reactors to scale up the system and kind of fine-tune the selected parameters like pH, retention time, and used chemicals. So, for example, pH seemed to have a quite notable impact on the metal extraction. Lower pH 1.20 seem to favor copper leaching, whereas at higher pH, closer to 2, more manganese was released into the solution. And for the piloting phase or the continuous phase, we needed to determine the retention time beforehand in order to estimate suitable flow rates. Kind of the good news in here is that all of our reactors, I mean different sizes that we have, they all have the same geometry, so they are easily scalable in that way. Although like scaling from 10 liter reactor to 100 liter reactor, several critical parameters like oxygen requirements, nutrient availability, and physical effects like mechanical shearing of the bacteria cells needs to be considered. For the piloting and the continuous phase run, we also tested the filtrability of the bioleaching slurry, and we noticed that there were some challenges in the filtration during the filtration. So there was no like a proper cake forming. It was more like a gel-like sludge. So this was something that we needed to tackle as well. And then the final stage. At the moment, we are running the continuous mode bioleaching pilot in a 100 liter reactor to produce solution to the next process steps, which are purification and precipitation. So for that, we prepared a 15 liter inoculum, and then it was added to the 100 liter reactor together with the modified 9K nutrient solution at 40-degree temperature with aeration of 20 liters per minute and mixing speed of 252 RPM. And at the ramp-up phase, the solid liquid ratio of the reactor was increased in stepwise in 4 batches and after to 12.5%. And after the target SL ratio was achieved, the reactor has been now running in it was run in the batch mode for 10 days, and then the continuous run was started. And then that will last 3 times HRT, so the total time will be 30 days. We have been now running it about 20 days, so we still have a little bit ahead of us. We are using calcium carbonate to maintain the pH at target 1.43, but it is also used to generate carbon dioxide in situ in the reactor. And some preliminary results indicate that the metal extraction efficiency of cobalt is around 95%, around 50% for copper, and around 70% for manganese. at the end, it turned out that the nickel content in the material is insignificantly low. So of course we are leaching nickel as well, and then recovering it in the precipitation mode, but that the concentrations are insignificant. So so far, this is the largest continuous mode bioleaching experiment that we have been running at VTT hydrometallurgical facilities. But I would say that the journey has just begun, and there will be many more. Yeah, so that was my presentation. Thank you.
Thank you very much, Tiina. That was really interesting. And now, last but not least, we come to the presentation of Douglas Pino Herrera. He's a researcher in process engineering, and he has been working at BRGM on the developing and upscaling of bioleaching processes in several European projects, in which he has been responsible for the bioleaching pilot operations. In these projects, he contributed to the development of models and process simulation for bioleaching processes, and lately his interests have been focused on developing a process eco-design approach to include not only technical and economic aspects, but also environmental impact evaluation, particularly through life cycle assessment tools on the development of emerging technologies. Okay, Douglas, ready when you are?
Yeah, do you hear me? It's fine?
Yes, everything works.
Perfect. So thank you very much for the presentation, and thank you for the invitation to participate in this webinar. So I'm going to be talking about bridging research and practice, particularly in the challenge in the design and the upscaling of bioleaching reactors. So after the excellent introductions for bioleaching of Päivi and Tiina, our colleagues from VTT, I just want to go a little bit more into detail of what happens in the bioleaching process and what is required for bioleaching to occur. So here we have like a typical bioleaching setup and reactor. Like we have cells, so bacteria, we have the sulphide mineral, and we have gas bubbles. And then we have a leaching solution containing different ions, nutrients, etc. Normally at a low pH. And the first thing that happens it's that we have if we have ferric ions in the system, these ferric ions are going to react to with the sulfide minerals and are going to reduce this ferric iron into ferrous iron. So you have the equation to the right of the slide, and then we will have this let's say production of reduced sulfur compounds. So this, which is represented here by the elemental sulfur. So actually, what bacteria are going to do is to regenerate the ferric ion to continue the reaction, and also to oxidize the sulfur compounds into sulfates. And to do this, actually, they are going to need oxygen, and they are going to do both. They're going to consume protons, so acidity from the system, but they're also going to produce this acidity from the sulfur compounds. So then, okay, we have different kind of mechanisms. So we can have non-contact and contact mechanisms to do that. So we can have the developing of biofilms, which are going to, bacteria or microorganisms are going to be attached to the system. And then actually for macrobial growth, many bacteria, most of them are the most used ones in bioleaching processes industrially, they need CO2. So CO2 in order to build biomass. And all these gases come of course from the air or the gas that we're feeding the system with, which is mainly composed with nitrogen, but also, of course, we have oxygen and CO2 in this air. And then we also have some processes like which are around these. So we have bubbles in the system. So we will have also water evaporation. So we have to be careful with this. And then also we may have some secondary processes like precipitation. This is the case, for example, for the gyroside, which also is going to take some iron 3. So ferric iron ions, and is going to combine with some cations and sulfates, and going to precipitate either on the surface of the sulfide minerals or in the system, also so these are things that we have to consider when we are designing reactors. So we can actually classify all these processes as the phenomena that I just explained. We can classify them in three big families. So dissolution, precipitation, and solid liquid mass transfer phenomena, gas liquid mass transfer phenomena, and biological oxidation. So these three processes in a reactor actually are going to be driven by physicochemical parameters of the reactor, which are pH, redox potential, surface tension, the concentration of the dissolved oxygen, or the viscosity of the system. And of course, these are going also to be driven by the hydrodynamic conditions, the mixing and the mass transport of the different compounds. And all these inside the reactor are going to be influenced by the solid composition, by the reactor design, and by the operating conditions we choose. So as you see, actually, it's a very complex system, which we have 3 phases plus the bacteria, and we have a lot of mechanisms that are intertwined and are going to be impacted one another. So all these parameters that we fix are going to define the physicochemical environment that ultimately controls the bioleaching reactions. So this is a big introduction for what I want to say is that why reactor design matters, because actually all this process that I just explained have some requirements. For example, the oxygen demand from iron and sulfur compound oxidations is going to be needing efficient gas liquid mass transfer. The solid dissolution reactors are going to be needing good solid liquid contact. The microbial activity in general and the growth actually are going to require stable temperatures and pH of the system. And for example, specifically the ferric iron regeneration is going to require proper mixing and proper residence times and all these kind of parameters that are important for the whole system. So the reactor designs actually it means that actually we need to control these transfer processes, which are the most important part of a bioleaching industrial process. And with transport processes, I mean mass transfer, so gas mass transfer, which is very important. We have understood this also from Tiina presentations, which actually they care about this process quite. They deem this process quite important. Then there is the mixing also, so the suspension of the solid particles, and the residence time also. So at the beginning of the webinar, we see the different kind of reactor configuration, which was a very good introduction for what I'm going to say now. Actually, in bioleaching, as also Päivi said, there are 2 main technologies or type of reactors applying: the heap leaching, which and the stirred tank reactor. In general, we choose one or another. Actually, there are also other configurations in between these two, as Päivi mentioned, but these are the most used. So in general, we're going to choose this thanks to the characteristics of the system. So maybe if we have some of the advantages and disadvantages, which are in the slide, if we want the heap leaching system is a low capex, low cost operation, let's say, suitable for low-grade ores, but has low kinetics and recovery yields, and also have a large footprint in the like physical footprint. And then for the stirred tank reactors, we have the robustness and the reliability, which are very high, with high recovery yields also, but we have more costly operations, and also it needs also more costly mineral processing steps before the bioleaching. So what is all this has to do with the scale up. So as you see, this is a very complex process. So usually when we study a bioleaching process, we start by shave flasks. So shave flasks typically are doing for screening tests. So we have several cultures. We need to see, for example, we want to see which one of these cultures are going to be used are going to be efficient in our system, are going to be able to extract the metals that we want. So we do this, we do shave flasks, we study the microbial activity, we study the microbial growth and these kind of things. The main challenges about this are actually that in this flask, actually we have a limited oxygen transfer. So the bacteria are probably going to be limited by this. I mean, the process is going to be limited by this. There is poor solid suspension because the agitation is generally not very efficient, particularly for heavy particles such as sulfide. That's why actually we choose always low pulp densities. So usually 1 to 3% by weight of solids in the system. And actually this may have an impact on the microbial growth. We know that bacteria may grow on it, but these are not the ideal conditions. But that's fine because then we can go to the lab scale reactors. And then we have a more controlled system. Then we have agitation and aeration in the system that are more efficient. And actually, we can increase the solid content to 5 to 10% in general. It can be a little bit more. And then we have a heat transfer system. So on the reactors, which will help us to understand better the system, to control better all these microbial reactions. So the advantages that we have for mechanical agitation that is improved for the solid suspension, and we have better temperature control and gas mass transfer. But we still have some problems. So when we increase the solid content, we will increase the oxygen demand, which is particularly when the sulfide content is high. And then we may have increase of power requirements, and we have the risk. We will try to go actually to fulfill this in the small reactors. We rapidly encounter risks of flooding the impeller, or poor gas dispersion, or some microbial damage inhibition because of the high velocities we need to increase all these parameters. So that's why actually what we generally do is once we study the system and we know that the efficient leaching is done in small reactors, we go to pilot scale reactors, go from 10 liters to several cubic meters and more. And then we can go to 10 to 30% solid, which are the real actually scale solid content in the pulp. And then these systems are more adapted to continuous operations. It's possible to obtain higher gas flows, but we need to be careful actually that the transfer in the system is still efficient enough. So we have a better evaluation of the oxygen demand and a more representative dynamics of the system. And this is the scale actually we usually use to obtain data for modeling, for environmental assessment, techno-economic assessment, and this kind of necessary steps in the industrialization of a process. So the challenges are still that we are increasing the system, so we're increasing actually probably the heterogeneity of the mixing and also of the gas dispersion. So this might be a little bit tricky. We need to be careful with this. It doesn't mean that actually for a small scale they work very well. When we have a bigger scale, we have to be careful that all the parameters are following the correct path to a bigger scale. And finally, when we get to the industrial scales, which is the continuous full-scale metal recovery, and we need actually all the processes that I already said, we already mentioned. Actually, we need to be sure that in industrial reactors they're correctly and proper managed. So in general, as a reactor size increases, the bioleaching system shifts from a reaction control to transport control processes. And actually this makes the reactor design critical. So now I will speak a little bit about the XTRACT project, which is actually something that this is a scale-up, but something that we're doing currently. So in the XTRACT project, the objective is to develop low impact technologies for metal recovery from low grades ores, mining waste and tailings, and difficult or uneconomic deposits. And the contribution of the project will be improved resource utilization, reduced mining waste, and lower environmental footprint of the metal extraction. In particular, there are many processes, many systems in the XTRACT projects. I'm going to talk about specifically the one that concerns bioleaching, which is investigated as an alternative extraction route based on microbial oxidation of sulfide minerals, recovery of metals from low-grade mineralization, and the potential in situ applications. And this research focuses on the microbial activity and iron oxidation, the metal dissolution kinetics, and the solution management and recovery. So there are 4 pilot cases. So the first pilot case is located in Germany. It's the Tellerhäuser deposit, which is a polymetallic sulfide mineralization, which contains zinc, iron, tin, and copper and indium. There are 4 partners: the Technical University of Freiburg, BRGM, GEOS, which is SME from Germany, and Saxony, which is the concession holder of the deposit. And the objective actually of these pilots are to establish and optimize the bioleaching technology, namely to test in situ bioleaching implement sustainable methods for the metal recovery, and save energy on this metal extraction process. So you can see a little flow like schema, actually a schema in the lower part of the reactor. We already done some reactor studies at lab scale and intermediary scale, particularly into BAF and BRGM, also GEOS on the metal recovery and reactive recycling part. And the idea here, we are working on reactors, but also mainly on columns, because what we need is the bio-oxidation of the iron. And then with this, actually, we're going to go to columns to mimic the in situ leaching, bioleaching process, and we're going to recover actually from these columns the metals, and we're going to recycle the iron, and so we're going to inject it in the system. So my last slide is the take-home messages on this scale-up design reactor and upscaling process. Is bioleaching is a complex multi-phase system. You are, I hope that at least you have understood this. There are biological, chemical, and transfer processes that interact with each other. Reactor design determines actually the process performance, and namely oxygen transfer, mixing, and solid suspension are critical for this process. And the scale-up changes the limiting phenomena. So we go from at the beginning, the small scale is more reaction limited, and then we go to mass transport limited phenomena. And the system become, as I say, it is something actually we need to be really careful when we go to upscale the processes. And then we have projects like XTRACT, which aim to bridge this gap, connecting laboratory studies with field application and check how all this is interconnected. Thank you for your attention. Some acknowledgements in the slide, particularly to the organizers of the webinar, my colleagues at BRGM, the Project XTRACT, and the European Union for the funding. And finally, just I wanted to do a little bit of advertising of 26th International Biometallurgy Symposium, which is going to be in France, in Orléans, organized by BRGM. So the submission actually for the abstract still open only for poster session. For oral sessions are already closed, but please, we invite you to Orléans, to France, to assist to this to talk, if you want to talk more about biohydrometallurgy, which is a super interesting subject. And yeah, so that's pretty much for me. Thank you very much. And if you have questions, please.
Yeah, sorry. Thank you, all three of you, for your presentations. I would like now to ask all the three speakers to turn on their cameras for the Q&A of the second block, which will be led by Päivi. We are a little late, so this is going to be a rather short Q&A before we then come to the open discussion.
Okay, thank you. There are some questions from the audience. So the first one is that what are the solutions provided so far for increasing the kinetics of the bioleaching? So it was clear that bioleaching processes are very low. So what are we able to do to increase the kinetics?
This is a question.
For both you, me? Douglas, perhaps you can go first, and then Tiina.
Okay, yeah, it is true, actually. This is the main thing that actually we always say about bioleaching compared to classic leaching, is that it's always a process which is always slower than the other ones. And the main thing is actually that it is the main thing that actually is going to slow down the process. It's not slow down, it's just like it happens at quite atmospheric conditions, mild temperature. And the thing that is slow in these conditions is actually the leaching part. I mean, the chemical leaching part, because bacteria actually are quite fast at oxidizing and regenerating the reactive. So what we usually do to increase this, but actually would increase the cost, so it's a trade-off as usual, is decreasing the particle size. As we decrease the particle size, we will have a faster leaching, and bacteria can keep up until a certain time, until a certain level, let's say. They are going to be able to keep up with the leaching. So we can do this, we can increase, but we have to be careful, because bacteria also are sensitive to things like metals in solution, like size particles. It can be so small, like particles as the bacteria, so we can have some interactions there that might be a little bit, let's say, yeah, it can go on the contrary to the normal bioleaching process. So this is one of the things that we usually do, but yeah, I think there's a lot of research to do about this area.
Okay, thanks, Douglas. There's also a question about the need for research on nutrient requirements of the used bioleaching bacteria. So is it easy to cultivate these microorganisms in bioreactors to high cell densities? Perhaps Tiina, you can take this question.
Well, I have to say I don't have that much experience it, but we have been working only very how to say, few bacteria so and microbes. So yeah, maybe that's something that's more like microbiological side of the field, I would say.
Douglas, do you want to add something? Thanks.
Yeah, maybe I can add that actually, in general, at the right conditions, these bacteria are not that hard to cultivate, and actually they are ubiquitous. They are all over the world. They are quite easy to find there where there are sulfides, and there is a little bit of water, and there's air. There are bacteria, bioleaching bacteria. So we can actually look for them. We saw some pictures actually on the presentations. I don't remember if it was the Päivi one or the Tiina's one, but we saw actually this drainage amino acids being produced, and then we saw these colors are a little bit this full of bacteria everywhere. So I think they are quite easy. And quite easy to maintain. They are very robust bacteria. So we can actually leave them, we can cultivate them, leave them aside for months, and then we can take them up, put them in the right conditions, and they're going to be still active after some time. So this is not a big issue for me. The cultivation is quite easy.
Okay, it seems that there are like a lot of questions. I don't know if we have time to go through everything. So what
Maybe
do you think, Ramona?
One last, and then we can go over to the big discussion round. Maybe a short one.
These are pretty long ones, but okay. So what is the strategy that you have used for upscaling to keep the hydrodynamics and other factors? And also perhaps this can be like widened to the question, like what kind of things do you need to take into account when you go from lab scale to the pilot scale? And both of you have done that in your research.
Yeah. Do you want to go first, Tiina, or yeah?
Yeah, well, I could maybe say that, well, compared to what you were presenting, those like cubic meters, we have the 100-liter reactor at the moment, but I would say that the physical things, like the mixing and aeration and things like that kind of are, at least from my point of view at the moment, are the things that I realize that are a bit different in the small scale than in the bigger scale.
That's right. To complete the answer, it is true that we have to look for the engineering parameters actually to keep this hydrodynamics and this oxygen mass transfer and CO2 mass transfer the same. So we know these parameters are the oxygen transfer coefficient, for example, the KLA, famous KLA that in process engineering is quite known. This parameter we try to keep. We calculate what is the consumption of the bacteria at small scale, like the maximum one, and we try actually to have in the bigger scale to have a transfer rate of oxygen that is at least as fast as the bacteria can consume this oxygen in the system. This is for the gas transfer, and the one for the mixing, as Tiina said, is actually we look at the power by unit of volume injected in the system. So we can also try to maintain this one, but we have to be careful because both of them, when we change both of them, there are a lot of things that change in the system, like hydrodynamics, like the distribution of the bubbles in the system and all these things. So these are the things that we have to really think about. And of course, this is the temperature, if it's homogeneous, if, yeah, how easy is to control, pH, these kind of things.
Okay, thank you. It seems that there are like a lot of questions, but perhaps you can reply to them in the chat or Q&A.
This would be very great. Maybe if you have later on the time to answer the questions in the chat. So we see it's a very, very interesting topic, and all the audience is very engaged in that. And I'm very happy that we can come to a very short, closing discussion round. So first of all, I want to thank all the speakers for their presentation, and I want to welcome all the speakers back to the screen. So if you can put all your cameras on, so then that we can see each other while we are discussing. And I also want to motivate the audience to put on their camera so that we can see who is here today. So yeah, so welcome back on screen. And now we are approaching our core question, which was how reactors for leaching and bioleaching are working, insights from research to practice. And the discussion will be now a little bit shorter. So we maybe have first a look on the gaps, what we are facing in research, and later on we are looking what the future might bring to us. So my first questions to the speakers would be, where do we still see knowledge gaps or limitations when it comes to reactor design, either for bioleaching or for leaching? And maybe Päivi, you can start to give a short feedback on this question.
Yeah, of course. In reactors, one of the things is like getting things mixed and getting them mixed with the like lowest possible energy consumption. So definitely, I think that that these kind of sustainability issues are there, like consuming as little as possible energy and so on. And then, of course, then especially with bioleaching, the thing is that quite often the materials are very low grade. So how to get these then like leached efficiently, perhaps even going to the higher temperatures and so on.
Okay. No. So do we see similar knowledge gaps in the leaching approach? Maybe from the leaching section, maybe Ben, do you want to go on?
Yeah, I mean, I think the sort of from the leaching without the bio component, I think that the issues are very much the same. Of course, it's about getting things mixed, making sure that there's, we're going for this more circular and green hydrometallurgical process. There's this quite famous paper from K. Leuven about the 12 steps to a green hydrometallurgy, so reducing energy consumption, reducing added chemicals and things like that. So I think that's the way that things are going, especially when we're thinking about the processing of secondary raw materials. So things like battery waste, these are okay, they're quite rich in metals, so they're similar to minerals and ores, but on the other hand, they come with their own set of issues as well. So I think, these kind of new materials, if you want to think of it that way, also have their issues when we're thinking about the downstream processing as well, filterability and separation techniques. So I think there's still a lot of targets for development in that respect. So still, we've got a very good basis from the historical, but these things are developing all the time. So I think there's still a lot of a bright future for hydro and biohydro applications.
Okay, yeah. but still a lot of gaps which we in research can follow. Maybe Tiina, do you like to tell us how it is in Metallico? So where have you experienced their knowledge gaps? Where I say, okay, for our reactor design here, it would be great to have this more knowledge.
Well, I would say that the engineering in the the reactor design and that, so I would say that that kind of was not maybe in the focus at the beginning of the project. So because we have been working only I would say the 10-liter reactors, so the continuous flow mode, bioleaching and so on. So I think we kind of understood that thing maybe a bit later phase in the project.
No. Yeah, sometimes it's like this. So maybe Anjay, coming to the another perspective, to the upscaling, we saw in your presentations a lot of big reactors and big sites. So maybe we would like to hear your estimation for the collaboration between research and industry. Where do you see, or where do you have experienced the benefits?
Okay. At the beginning, let me please to come back to the construction of the reactor in our part of METALLICO project. As you know, we treated sponge, which was iron arsenic alloy. So we have to prepare a reactor which will be constructed on the way that gives us safety, because generation of hydroarsenide is very probable in such a processes. So it was our main problem we solved successfully. And
okay. Mm-hmm.
Going to the cooperation between science and industry, we got in our consortium representatives of smelting sector like Cobreras Cruces, and this very activity in our cooperation or cooperative work gives a clear view that for both sides, scientists and industrial managers and engineers, this cooperation is fruitful. Thank you.
Great. Yeah. So maybe because we are a little bit out of time, we go over to the last wrap-up question. This means I want to hear in just one sentence from all the speakers of you one question and one answer to the question. And the question would be, what should be the priority for reactor design and leaching or bioleaching over the next 5 years? So what are your wishes for the next 5 years? So maybe Douglas, do you like to start?
Yeah, it's a good question. I would say that actually in terms of bioleaching, which is my domain, I think one of the things that I think would make advance more this scale up processes from because we have have some projects on small scale, but they don't go to full scale. And actually, what I would like to see in the next 5 years is actually the supplying all these concepts, particularly in the smaller scales, because we know it works very well in small scales, and when we go to bigger scales, all these concepts of engineering, which are very important, which are very we need to keep them in mind, and then we fill the gap between actually the laboratory and the industry through actually multi-disciplinary work between microbiologists, process engineers, chemical engineers, system engineers, and other things. I think this is something which is very important for me and going through to the project to the full scale. Yeah.
Okay. Thank you very much. So maybe Ben, do you want to add from the teaching perspective? So what is your wish for the next 5 years?
Yeah, I mean, I would echo what Douglas said. I mean, I think the level of education for the reactor engineering and process design, so it's very integrated, would be very beneficial. I mean, there's knowledge there, but trying to link those together. And I think there's been some discussions. Okay, we have reactors on the large scale, which are very effective, but can we actually scale those down? So then, to the lab scale, that then we could, ease the scale up if we're looking at different processes. So there's some discussion about that I've heard, but I think making sure that there is this good coordination on a sort of educational level, that that everybody gets the basics of process design and reactor design, which will really inform their future careers going forward, would be, I think, very useful.
Great, thank you. Who likes to add? Maybe Mateusz, we didn't.
So from my side, I would say that make this path between, let's say, scaling up, commercialization, PFS study, and launching the final installation much shorter would be most beneficial for the all future perspective projects in Hydromet shortly. Thanks.
Thanks. No, maybe Joanna, you have something to add to that. What is your wish? Okay. I think there is an issue with your microphone. Maybe Päivi.
Yes, I have.
Okay.
I have a microphone. Leaching reactor for the leaching is very interesting subject. I think it's a long topic for maybe next webinar.
Thank you very much. And then Päivi, from your perspective in bioleaching, what is your wish?
I just wanted to emphasize that the important thing is that the parameters are right. Even in the small scale, it's possible to get really valuable results if the parameters are relevant for like real scale applications. So definitely it's worthwhile to scale up, but also the small scale experiment should be done with the industrially relevant parameters.
Yeah. These are very, very nice closing words. Very nice. So yeah, then I would say thank you very much for this discussion. As we are already a little bit late, I would stop right here. Nevertheless, I will share the screen for the last and closing words. So thank you. This was what I wanted to say. It has been, a lot of work behind the scenes and a lot of support that we got. And first of all, I want to thank again all the speakers for their time, for their effort, also for this last nice discussion round. Also a big thank you to the sister projects, Theta-led, Respect and Extract, for coming here to the METALLICO webinar. I also want to thank you for your experience, but also from the support from your communications partners for promoting this webinar. This really helped us very much. At this point, I also want to thank the Cluster Hub who were also sharing this event. And yeah, last but not least, if you like to learn more about what METALLICO is doing, just watch the latest video of our first webinar. It was about how life cycle assessment can support innovations in minerals metals production. And of course, like Douglas already introduced to you, if you like to learn more about bioleaching, there is this upcoming symposium this autumn. So if you want to go there and have a poster, just go there. Yeah. So thank you very much.
I just put the website on the chat if you want to see have more information on the symposium. Thank you.
Perfect. That's great. Yeah. So thank you all, and I wish you all a nice afternoon. And